Nematophagous fungi. Soil nematodes are small roundworms, some of which are parasitic on plants and animals and may cause severe damage to crops and livestock. A group of soil fungi are natural enemies of nematodes. These fungi have developed different mechanisms of capturing, killing and digesting their prey. The fungi have the ability to change their morphology so that nematodes may adhere to them or be captured in mechanical traps. For instance, nematodes are captured in constricting rings, or they are held fast by sticky branches. A captured nematode is inescapably gripped by the fungus. The nematophagous fungi capture nematodes either with special mycelial structures or with their spores, as in the endoparasitic fungi.
Some fungi attack nematodes with motile zoospores. Others infect their hosts with adhesive conidia. Endoparasitic fungi often live as obligate parasites in the soil.
The uniflagellate zoospores of Catenaria anguillulae are 2 to 3 micrometers wide and move rapidly in the water film on an agar surface. They are chemically attracted to nematodes and aggregate at the mouth or other natural orifices. Here the nematode Pangrellus redivivus has been immobilized by cold treatment. After contact, the zoospores produce an adhesive, withdraw their flagella, form a cell wall, and become attached to the surface of the host. From the encysted zoospore, a germ tube is formed that penetrates the nematode. The same situation seen in the scanning electron microscope. A few zoospores still have their flagella, while others are already encysted. Following penetration in the mouth region, trophic hyphae develop within the nematode and digest its contents. Segments of the hyphae develop into zoosporangia 10 to 12 hours after infection. They subsequently mature and produce evacuation tubes. A few minutes after the zoospores have differentiated inside the zoosporangia, they are released. The release is shown here somewhat faster than real time. The zoospores can now attack new nematodes. Trapping strategy is used by Nematoctonus concurrens with the aid of its hourglass-shaped knobs, which are equipped with an adhesive substance. The head of a nematode is here seen in contact with a trap, and the nematode is attached to it. In spite of its efforts, the nematode is firmly gripped. The motility of the nematode decreases after the fungus has penetrated its cuticle. After some time, the nematode is completely filled with trophic hyphae. In the head region, lower right, the trap from which the lethal infection started can be seen. The fourth example of a nematode trapping fungus uses constricting rings. The rings of Arthrobotrytis dactyloides consist of 3 cells attached to the mycelium by a stalk. When touched, each of the 3 cells suddenly swells, forming 3 spherical structures. In the laboratory, here in Dactylaria brocopaga, the swelling of the 3 cells can be induced by heat treatment. Note the rapid cytoplasmic movements near the internal surface of these cells. Sometimes nematode larvae, for example of Aphelenchus avini, may enter a trap without causing the trap to close. After closure of the trap, hyphae from the swollen ring cells grow into the captured nematode. The digestion process is similar to that of other nematode-trapping fungi. Nutrients are transferred to the fungus.
Some fungi possess other capture mechanisms. Verticillium suclosporium infects nematode eggs by using its hyphal tips. Pleurotus austriatus produces droplets on its hyphae which are highly toxic to nematodes.
This group of fungi do not form special infection structures with which to attack nematodes, but use their hyphal tips. The growing mycelium cannot attack a moving nematode, but invades the eggs of cyst and root knot nematodes. These are eggs of the sugar beet nematode Heterodera schachtii, a serious parasite. Here, a non-infected egg with a fully developed larva. The hyphal tips of Verticillium suclosporium may enter the egg through the egg shell. At the penetration site, the hyphae may form an aprosorium-like swelling. After 3 weeks, disorganization of the internal structure is evident. New hyphae penetrate through the eggshell and form a new mycelium outside the host. An infected egg at an early stage, and here at a later stage when hyphae are visible inside. This scanning electron micrograph of a crushed egg allows a view of the dense mass of hyphae inside the shell. The final example of a fungus with nematode-destroying capability is Pleurotus austriatus, with its toxic droplets. The fungus has developed a strategy to immobilize its victim before it is invaded by the fungus. The toxin is released from short hyphal stalks, which are thickened apically. When a nematode touches these droplets, the toxic effect is reflected in the animal's behavior. After only a few minutes, the nematode becomes immobilized and then distorted. The poisoned nematode elicits chemotropic growth of new hyphae, which enter the worm through its mouth. Some hyphae enter the nematode directly through the cuticle, form an infection bulb, and, from the bulb, trophic hyphae. The nematode dies. Irrespective of the infection method, the result is always the death of the nematode. The ability of nematophagous fungi to attack nematodes and use them as nutrients may have a regulatory effect on nematode populations. It is obvious that these fungi have a potential to be further developed for biological control of nematode parasites on plants and animals.